Cell, the unit of life. So first we study about cell, which is the structural and functional unit of life. It was first discovered by Anton von Leeuwenhoek, first live cell. Then Robert Brown discovered nucleus. Now majorly there are two types of cells. There are multicellular and unicellular cells. Now multicellular are the one which have many cells and can perform the functions in a coordinated manner, whereas unicellular have independent existence and perform essential functions of life like digestion, respiration on their own. Now next we have the size of cell, which is different depending upon the structure of the cell. So first of all is the largest cell, ostrich cell. Smallest is mycoplasma, which is 0.3 micrometer. Longest is the nerve cell, and lastly we have RBC, which is the cell of ours, which is basically RBC having 0.7 micrometer in diameter. Okay, next we have another important thing to note here is about the cell theory. The cell theory formulated by Matthias Claden in 1838 was a German botanist who told that all plants and animals are composed of cells, and this cells forms the tissue. Next we have Theodor Schwann, who was a German zoologist in 1839. He studied the plant and animal cell. The plant cell consists of a cell wall, which is a unique character to it, but the animal cell is covered by a plasma membrane. Okay, next he gave a hypothesis. What was the hypothesis? The hypothesis was that plants and animals are composed of cells. So basically plants and animals are composed of cells and its products of cells. All cells arise from pre-existing cells. All living organisms are composed of cells and its product. So this is basically the cell theory. What does the cell theory states? The cell theory states that all cells arise from pre-existing cells and all living organisms are composed of cells and are products of cells. Next, we come across prokaryotic cell. It includes blue-green algae, bacteria, mycoplasma, pleuropneumonia-like organism. The prokaryotic cell does not have a well-defined nucleus, and membrane-bound organelles are also absent. The nuclear membrane is again absent, and the genetic material is basically naked. Now, in genetic material, we have a genomic DNA and we have a plasmid. The genomic DNA is single circular DNA. The plasmid is a small circular DNA, which is an extra DNA which has unique phenotypic characters, that is, provides unique phenotypic characters such as resistance to antibiotics. Obviously, this is a naked DNA. Next, the prokaryotic cells have cell wall except mycoplasma. Okay, they also have majorly three layers of the cell envelope: outermost glycocalyx, whether it be tough or loose. If it is tough, known as capsule; if it is loose, known as slime layer. And then we have the cell wall, which provides structural support. Then we have the cell membrane, which helps in elongation or extension in blue-green algae. So cell membrane is there. Cell membrane is selectively permeable. Now in blue-green algae, there is extensions of the cell membrane which are known as chromatophores. So chromatophores are extensions of the cell membrane in blue-green algae which contains pigments. Very important. In blue-green algae, the cell membrane has extensions or elongations known as chromatophores which contains pigments. Next we have enfoldings of the cell membrane. The enfoldings of cell membrane is known as mesosome, which is in the form of tubules, vesicles, lamella, and the major function of cell wall formation, DNA replication, secretion process, and I think respirations. Majorly four major functions. What are they? Obviously they will form the cell wall, respiration, DNA formation, and secretion. Next we have surface structures, which includes flagella, pili, and we have the fimbriae. Flagella is having three major parts: the filament, hook, and the basal body. It's for motility. Pili are made up of pillin proteins, and fimbriae helps in attachment to the rocks. Okay, next we have ribosomes, which is present in the cytoplasm 70s. So prokaryotic only have 70s in the cytoplasm subunit, 30s and 50s, and these are made up of 15, size is 15 nanometer by 20 nanometer size. Obviously the ribosomes are site for protein synthesis, so there would be protein synthesis in these ribosomes. Now many ribosomes together come and form polysomes or polyribosomes. I am repeating it again. Ribosomes, we all know, were discovered by George Palade, and ribosomes are present in the prokaryotic cell, which is only single in the cytoplasm. That is 70s having two subunits, 70s having two subunits, 50s and 30s, and the size are 15 nanometer by 20 nanometer, and the site of protein synthesis. Several ribosomes transform into forming polysome, which translate mRNA into proteins. Next, we have inclusion bodies, which are basically reserve materials which are membraneless and flies freely in the cytoplasm. So reserve food material which are membraneless because they are present in prokaryotes and live freely in the cytoplasm. Gas vacuoles. Where are gas vacuoles? Gas vacuoles are found in blue green, purple green, and photosynthetic bacteria. Now inclusion bodies examples majorly are phosphate granule, glycogen granule, and cyanophysin. So phosphate granule, glycogen granule, and cyanophysin granule. Next we have, based on gram staining by scientist gram viz, if it is present then gram positive, absent then gram negative. Okay, some important notes you to remember is that anything less than a complete structure of cell does not ensure independent living. Cell shapes vary with their function. So if the cell shape is different, it has different function associated with it. Organization of all prokaryotes are similar, but shapes and function vary. So basically in prokaryotes the organization is almost similar, but the shapes, because when the shapes vary, the cell shapes vary, so the functions also vary. Bacterial flagella is the extension of cell wall. So the flagella which helps in motility is extension of the cell wall. Next we have prokaryotic cell, which includes the protists, plants, animals, fungi. It has membrane bound organelles present, and obviously the nucleus is well developed with a nuclear envelope. The plant cell has cell wall, plastid, and a large central vacuole. So what does the plant cell have which the animal cell does not have? It has a large central vacuole, it has cell wall, and it has plastid. Animal cell has centriole, which helps in cell division, which forms the centrosome. So very important point to note is that plant cell have cell wall, plastid, and large central vacuole, whereas animal cell have centriole. Cell membrane is composed of proteins and lipoproteins, that is lipids. So the cell membrane, outermost cell membrane, which we also studied in prokaryotics, here it is composed of proteins and lipoproteins or lipids. The cell membrane is also composed of proteins, carbohydrates, and cholesterol, which is very important to note. So the cell membrane consists of majorly four things. What are they? It consists of proteins, lipids, that is lipoproteins, carbohydrates, and cholesterol. Okay. Now the lipids in RBC and the proteins in RBC, the percentage is given in NCERT, that is lipid is 40% and protein is 52%. Okay, so basically lipids are 40%, so lipids are 40% and proteins are 52%. I'm repeating it again. In RBC, the cell membrane, the lipids are 40% and proteins are 52%. The proteins is majorly of two types: the integral protein, which is partially buried or totally buried in cell membrane; peripheral protein, obviously peripheral, which lie on the surface of cell membrane. Lipids are arranged in a bilayer, non-polar tail of saturated hydrocarbons. So the tail is non-polar, and the non-polar tail of the saturated hydrocarbon is protected from the aqueous environment or from water to prevent it from wetting. Next, we have the fluid mosaic model given in 1972, which states the quasi-fluid nature of lipid enables them to slide, or you can say for the lateral movement of proteins within an overall bilayer. Because of the quasi-fluid nature of the lipid, it enables the lateral movement of proteins with the overall bilayer. Fluid nature depends majorly on many things, that is cell growth, formation of intercellular junctions, secretion, cell division, endocytosis. So fluid nature depends on formation of intercellular junctions, cell growth, endocytosis, and the secretion and cell division. Obviously, the fluidity nature depends on cell growth. It depends on the intercellular junctions. If they are present, it will become less. It depends on cell division. Obviously, it depends on secretions. You can just think, yes, it does depend. It depends on endocytosis. ATP is used in active transport when there is low concentration to high concentration transfer. In osmosis and diffusion, there is just high to low concentration, or in passive transport. So there is no active energy required, but in active transport, ATP is required from low to high concentration, like sodium potassium pump is a very good example of it. Next, we have the cell wall, which is basically the giving shape and protection and helps in cell to cell interaction in the eukaryotic cells. Okay, so basically the cell wall which provides shape, protection. So the cell wall provides shape, protection. It was not providing cell to cell interaction in prokaryotes, but in eukaryotes it provides cell to cell interaction, and it also provides the barrier to undesirable proteins. So majorly four functions: it provides shape, protection, barrier to undesirable proteins, and cell to cell interaction. Now there are different cell walls. We have primary cell wall, secondary cell wall on the inner side of the cell, and the middle lamella made up of calcium pectate, which glues the different cell. Very important to note is that the middle lamella is made up of calcium pectate. Okay, so the middle lamella is made up of calcium pectate. The middle lamella is made up of calcium pectate, which glues the different cells. Algae made up of cellulose, hemicellulose, galactans, and calcium carbonate. So middle lamella made up of calcium pectate, whereas algae has majorly four composition in its cell wall. The middle lamella made up of calcium pectate glues the different cells. Cell wall and middle lamella traversed by plasmodesmata, connecting the cytoplasm of the neighboring cells. Matlab, cell wall and the middle lamella, which are joined together. Traverse means joined together. The cell wall and the middle lamella. We have cell wall, then we have middle lamella. These both are connected together, joined together by plasmodesmata, which connects the cytoplasm of the adjoining or the neighboring cells. Next, we have endomembrane system in the eukaryotic cells, which includes endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. So the endoplasmic reticulum are tiny tubular structures. Endoplasmic reticulum are tiny tubular structures which are scattered in the cytoplasm. Divides into inside endoplasmic reticulum, which is luminal, and outside endoplasmic reticulum or extraluminal in the cytoplasm. Endoplasmic reticulum shows ribosomes attached to the surface, smooth and rough endoplasmic reticulum. The rough endoplasmic reticulum helps in protein synthesis and secretion, where smooth endoplasmic in synthesis of lipid and lipid-like steroidal hormones. The rough endoplasmic reticulum cells actively involved in protein synthesis and secretion. It is extensive and continuous with a outer nuclear membrane. So the rough endoplasmic reticulum is extensive and it is continuous with the outer nuclear membrane. Okay, so endoplasmic reticulum is what it is basically. Endomembrane system is endoplasmic reticulum, which is tiny tubular structure scattered in the cytoplasm. Inside it is known as luminal, outside extraluminal cytoplasm. Next, we have Golgi apparatus. I already told you, which was discovered by Camillo Golgi in 1898. These are reticular structures near the nucleus, and these are concentrically arranged near the nucleus with distinct cis phase, convex, forming, and trans phase, the concave or the maturing phase. So basically, Golgi apparatus by Camillo Golgi 1898, reticular structures near the nucleus made up of concentrically shaped nucleus with distinct cis phase or the forming phase and trans phase, the maturing phase. The Golgi apparatus consists of flat disc-shaped sacs or cisternae 0.5 to 1 micrometer in diameter. The Golgi apparatus consists of flat disc-shaped cisternae 0.5 to 1 micrometer. So Golgi apparatus consists of flat disc-shaped cisternae made up of flat disc-shaped cisternae 0.5 to 1 micrometer in diameter. Functions of Golgi apparatus is helps in packaging of materials, important site for the synthesis of for the formation. Golgi apparatus helps in packaging the material, important site for the formation of glycoproteins and glycolipids. Lysosomes are membrane bound. Lysosomes are membrane-bound vesicular structures formed by packaging in the Golgi apparatus, rich in enzymes, hydrolase, lipases, proteases, carbohydrates at acidic pH. So basically, lysosomes vesicular structure formed by packaging of the Golgi apparatus. Hydrolases consist of lipases, proteases, and carbohydrates at acidic pH. Very important to know is that there is what acidic pH. Okay. Now the carbohydrates are capable of digesting proteins. So the carbohydrates is capable of digesting proteins, nucleic acid, carbohydrates, and lipids. So carbohydrates is capable of digesting proteins, nucleic acid, carbohydrates, and lipids. Now vacuoles are membrane-bound space found in the cytoplasm bound by a single membrane. So vacuoles are bound by a single membrane called tonoplast. In plant cell, it occupies 90% of the volume, and tonoplast facilitates the transport of the cell against the concentration gradient. So tonoplast is the single membrane found in the vacuoles. The amoeba has contractile vacuole for osmoregulation and excretion, whereas protists have food vacuoles. Amoeba has contractile vacuole for osmoregulation and excretion, and protists had food vacuole. The vacuoles contains water, sap, excretory products which are not useful. Next, we have come across mitochondria, which is obviously the site of aerobic respiration, powerhouse of the cell. Mitochondria, we all know, divides by fission. Now we have outer mitochondrial membrane and inner mitochondrial membrane. The outer mitochondrial membrane forms the limiting boundary, and the inner mitochondrial membrane is filled with matrix. So outer mitochondrial membrane is forming the limiting boundary. So outer mitochondrial membrane forms the limiting boundary, whereas inner mitochondrial membrane is filled with matrix. Now the shape of mitochondria is saucer shaped or cylindrical shaped, diameter 0.2 to 1 micrometer in diameter. Length is 1 micrometer to 4.1 micrometer. Number of foldings called cristae towards matrix. Function is increased. So function of cristae is increased surface area for absorption. Matrix possesses single circular double-stranded DNA, few RNA molecules, ribosomes 70S. So the matrix possesses the single circular double-stranded DNA, few RNA molecules, ribosomes which is 70S, And so mitochondria in prokaryotes is 70s. Ribosomes. Plastid is found in plant cell and euglenoids. It is seen under the microscope majorly three types: chloroplast, chromoplast, and leucoplast. Chloroplast obviously contain chlorophyll and carotenoid pigment, trap light energy, photosynthesis, and they are found in the mesophyll cells of the leaves. The chloroplasts are disc-like, so they are disc-like, lens-shaped, spherical or ribbon-like. So four major shapes: disc-like, lens, which we wear lens-shaped, spherical or ribbon-like. So ribbon, which we use to tie. So ribbon-like, lens-like, spherical, and disc-like. The width is 2 to 4 micrometer and length is 5 to 10. Simply remember 510. So 5 to 10 micrometers and width is 2 to 4. Just you multiply it, like 5 to the 10 and 2 to the 4. That's 1 per cell chlamydomonas or 20 to 40 cell in mesophyll. Chloroplast is double membrane. Outer membrane is more permeable. Inner membrane is obviously less permeable. The inner membrane has stroma, which contains thylakoids, and these thylakoids are arranged one on top of each other, forming the grana. The stromas contain single, double standard circular DNA, ribosomes, and enzymes for synthesis of carbohydrates and proteins. So there is ribosomes are present 70s only in the chloroplast. So in mitochondria and also in the chloroplast 70s ribosomes are present in prokaryotes. So I already told you stroma contains small circular DNA, enzymes for synthesis of carbohydrates and proteins, and ribosomes, which is 70s. Next we have chromoplast, which is fat soluble carotenoid pigments like keratin, xanthophyll. So fat-soluble carotenoid pigments like keratin and xanthophyll give red, yellow, orange color. Leucoplast, a colorless plastid, three types: a myeloplast, hyaloplast, and a neuroplast. A myeloplast, how to remember these? See how I remember. A myeloplast is for storing carbohydrates and fats, and hyaloplast for oils and fats, and aleuroplast for proteins. So simply remember hyaloplast. I comes in hyaloplast, so I oils and fats. A myeloplast, AMY. So just remember carbohydrates, HY. So YY. So carbohydrates, a myeloplast, and left with proteins. So how will you remember? Aleuroplast, proteins having E. There is no E in a myeloplast and hyaloplast. So proteins has E and aleuroplast also has E. Okay. Next we have the ribosomes, discovered by George Palade 1953. Sedgwick's unit measure of density and size, composed of RNA and proteins. So eukaryotes 80s, 60s and 40s, prokaryotics 70s, 30s and 50s, 50 nanometer by 20 nanometer size of 70s. Cytoskeleton are filamentous proteinaceous structures consisting of microtubules, microfilaments, and intermediate filaments. Function is motility, maintenance of cell shape, and mechanical support. Next, we have cilia and flagella. Cilia and flagella are hair-like outgrowths of the cell membrane, and these emerge from basal body. So see, they are hair-like outgrowths of cell membrane emerge from centriole-like structures called basal body. They are covered by plasma membrane. Core called axoneme passes number of microtubules running parallel to the long axis. Axoneme has 9 doublets of peripherally arranged peripheral microtubules and a pair of centrally located microtubules. 9 plus 2 array central tubules connected by bridges and enclosed by central sheet connected to one of the tubules of each peripheral doublets by radial spoke. Let me explain you. Cilia are basically small and they work like oars because movement of the fluid, either the surrounding fluid or of the cell. So cilia are very small, they work like oars and they cause movement of the cell or the surrounding fluid. Flagella obviously is larger, help for motility or cell movement. Bacterial flagella, prokaryotic bacterial flagella is different structurally from the eukaryotic one. Now what I read here was that basically it is hair-like outgrowths of cell membrane and emerge from centriole-like basal bodies, centriole-like structures. So centrioles, I will teach you ahead, the centrosomes have centriole which contains basal bodies, and the core is known as axoneme. Okay, next we have the axoneme, which possesses number of microtubules which run parallel to each other to a long axis, and it has 9 doublets. So see, these are the 9 doublets. Wait, 1 2 3 4 5 6 7 8 9. These are the 9 doublets of radially, so this is radially arranged peripheral microtubules. So these are the peripheral microtubules. You have marked peripheral microtubules, and a pair of centrally located microtubules. So this is a centrally located. This is peripheral, means outer, means peripheral microtubules, and this is centrally located microtubules. It is having 9. This whole is 9, and this is 2. 9 plus 2 array, which is enclosed by a central sheet. This is the central sheet. This one is the central sheet, and which is enclosed by the central sheet connected by the bridge. This is the bridge. See, this one connected to one of the tubules of each peripheral doublets by a radial spoke. So this is the radial spoke. This one I'm explaining. 9 plus 2 array. This outermost is the plasma membrane. Now these are the 9 doublets. 2 is there, so 9 plus 2 array out to 9 peripheral microtubules, 2 centrally located microtubules, and then we have this sheet-shaped structure which is looking. This is known as the central sheet, and this is the bridge which is connecting them, and these ones are the radial spokes. Okay. Next we have centrosomes and centrioles, which are surrounded by amorphous pericentriolar materials. So surrounded by amorphous pericentriolar materials are centrosomes and centrioles. Okay. So the centrosomes and centrioles are surrounded by amorphous pericentriolar materials. The centrosomes consist of 2 cylindrically arranged structures called centrioles. They are made up of 9 evenly spaced peripheral fibrils made up of tubulin protein. Both lie perpendicular to each other. It's like a cartwheel organization in centrosomes and centrioles. 90 degree, they are 90 degree, and they are 9 evenly spaced peripheral fibrils made up of tubulin proteins. Each peripheral fibril is a triplet. Very important. Each peripheral fibril is a what? It is a triplet. Okay, it is a triplet, and the central part of the proximal centriole is proteinaceous called hub, which is connected with the tubules of the peripheral triplets by radial spokes made up of proteins. Centriole form the basal body of the cilia and flagella and spindle fibers that give rise to spindle apparatus during cell division in animal cell. So basically, in centrosomes and centriole, they are spaced evenly, 9 of them, peripheral fibrils made up of tubulin protein, cartwheel organization. Each peripheral, one peripheral fibril, peripheral because outer, and one peripheral fibril consists of triplets, and central part of the proximal region of the centriole is proteinaceous called hub, which is connected with tubules of the peripheral triplets by radial spoke. So like this, there are also radial spoke in them. The centrioles forms the basal body of cilia and flagella and spindle fibers. So centriole क्या बनता है? It forms the basal body of cilia and flagella and spindle fibers So the centriole forms the basal body of cilia and flagella and spindle fibers that give rise to spindle apparatus during cell division in animal cell. Next we have nucleus, which was discovered by Robert Brown. Material of nucleus called chromatin by Fleming. Nucleoli are spherical bodies. Nuclear envelope has two parallel membrane with a space between them called the perinuclear space, which is 10 to 15 nanometers. So very important, the perinuclear space is 10 to 15 nanometer, which forms a barrier between the materials present inside the nucleus and that of the cytoplasm. So the perinuclear space forms a barrier between the materials present inside the nucleus and that of the cytoplasm. It is 10 to 15 nanometer in size. Next we have the outer layer of the nucleus remains continuous with the endoplasmic reticulum. So outermost layer of the nucleus is continuous with the endoplasmic reticulum. It bears ribosomes, the nuclear pores, which are minute pores through which the movement of RNA and protein molecules takes place in both directions. So nuclear pores are minute pores through which the movement of RNA and proteins both take place in both direction in the nucleolus and cytoplasm. The nuclear matrix composed of nucleoplasm, which consists of nucleolus and chromatin. The nucleolus is continuous with the rest of nucleoplasm. The nucleolus is a non-membrane bound structure, site for active ribosomal RNA synthesis. So where does active ribosomal RNA synthesis occurs? It occurs in the nucleolus. Chromatin contains DNA and basic histone proteins. So chromatin contains what? DNA and basic histone proteins, which is basic histone proteins called histone, non-histone protein and RNA. The nucleoli are spherical structures present in the nucleoplasm. Now you can see the basic structures. This is the above part. Okay, wait. This is the above part. 1, 2, and the middle part is the this part is the centromere, and these are disc-shaped structures which are known as kinetochore. The centromere holds two chromatids of a chromosome. Now it can be metacentric, submetacentric, acrocentric and telocentric as shown in the diagram. Next we have small fragments which are known as satellite, appear in few chromosomes have non-staining secondary construction. So small fragment called satellite appear in few chromosomes, have non-staining secondary construction. Microbodies are minute vesicles, membrane bound, contain various enzymes present in both plant and animal cells. So microbodies are membrane bound minute vesicles, contain various enzymes present in both plant and animal cells. Some important notes are: first of all, the physiochemical approach to study living organism is reductionist biology. So physiochemical approach to study the living organism is known as reductionist biology, which is an important previous year question. And G.N. Ramachandran discovered the helical structure of collagen and published in the Nature 1954. So who published the helical structure of collagen? G.N. Ramachandran discovered in 1954. It was published in the Nature. Thank you.